My name is Barbara Drasler and I'm a postdoctoral researcher at the University of Fribourg in Switzerland. I will present a study on long-term exposure of 3D human airway epithelia to quartz. This study has been performed within the scope of the EU-funded project PETROLS. The mission of the project is to deliver advanced and realistic tools and methods for nanomaterial safety assessment. One of the goals of the project is to produce lung culture models for nanosafety evaluation, and this is what I will also present in the talk today. The tested material selected in the study was silica quartz, in particular DQ12. This is a reference material for induction of lung inflammation. The size of the particles ranges from nano to micro scale. DQ12 particles have been shown to induce inflammation and fibrosis in rats in vivo studies. In this study, we used commercially available tissues of human bronchial region derived from healthy and asthmatic donors. We added human macrophages onto the tissues and exposed the tissues to DQ12. The exposure was performed at pseudo-air liquid interface. This means that a thin layer of dispersion was applied onto the tissues. We exposed the tissues to either a repeated long-term doses of DQ12 or with a single high dose. In both the cases, it resulted in 10 micrograms of DQ12 per centimeter square over 10 days. We visualized the particles with the corresponding doses using TEM, and this confirmed a non-homogeneous size distribution of particles with predominantly the size around 5 micrometers. Enhanced dark-field imaging revealed that particles came in contact with cells in both healthy and asthmatic tissues. Spectral analysis confirmed that these shining dots are indeed DQ12 particles, as the spectra matches to that of DQ12 alone. And also, we observed that the distribution of particles within the tissues was unequal. This means at some cases there was no particles, in some parts there were few of them, and some parts were packed with DQ12. There was no effect observed on cell viability after exposure to DQ12 in both healthy and asthmatic tissues. Also, there was no inflammation observed after 10 days of exposure to DQ12. Here I'm showing the results for healthy tissues only, as analysis of asthmatic tissues is still in progress. But we can see in asthmatic tissues that morphology of the tissues remained intact upon exposure to DQ12. We can conclude based on the data that DQ12 particles did come in contact with cells. As of now, we can say that it did not affect tissue morphology, cell viability, and it also did not induce pro-inflammatory response. We are looking at other endpoints, such as profibrosis and barrier integrity. We also are investigating the role contribution of mucus in these particle-tissue interactions. I would like to thank the team at the University of Fribourg, and I would like to thank the whole PETROLS consortium, in particular the partners of Work Package 3. Thank you very much for your attention.